Dr. Orville Baldos. He'll be giving a presentation, his updates on native Hawaiian ornamental research. Dr. Baldos is an assistant researcher in sustainable ornamental production at the Department of Tropical Plant Sciences, University of Hawaii at Manoa. His research interests include native Hawaiian plant materials development for landscape and ornamental use, screening and selection of non-invasive ornamental species, and development of sustainable ornamental plant production and landscape management practices. Dr. Baldos, thank you for joining us today.
Thank you, Glenn, for the introduction.
You're welcome.
I'm going to share my screen now. Okay. All right, so today's talk is just some updates on native Hawaiian ornamentals research. We'll specifically talk about these two species. First would be talking a lot more about peperomia or Ulaua vainui, and then some short updates on Pawhiaka. So Ulaua vainui, they are species in the genus Peperomia. In Hawaii, we have 25 species, 2 are native, and then 23 are endemic. Ulaua vainui have varying leaf shapes, sizes, and colors, which has the potential for selecting plants for indoor use. They're also a native succulent, so they can actually survive with drying out, periods of drying out. So this photo here, I just saw an article back in 2018 about peperomia being a hot plant. And even though this is 2019, it's still a hot plant. People are still looking for peperomia plants. So that's one of the reasons why I was looking at peperomia as a potential ornamental native. So the objectives of our research program was basically to collect germplasm from nurseries, also from in situ, so in the wild, with proper collections. And we establish horticultural protocols, and then we evaluate these plants under indoor conditions. So some updates on germplasm collection. So this year we fortunately had a trip to Maui before the lockdown, and we collected these two species, Peperomia macriana and Peperomia blanda. So these will be in the pipeline in terms of evaluation under indoor use. Last year, we collected a bunch of plants from the Big Island, and we found Peperomia hypoluca and this selection of Peperomia kukiana as potential selections for indoor use. So we're currently in the process of propagating these, increasing these, and then hopefully once we get the numbers, we can put them under indoor conditions and see if they last, or if they are good candidates. On the propagation side of things, we discovered turfis. So turfis is basically a calcium clay, so it's baked clay that's used actually in sports fields and also for turf applications. But I found this one in a succulent forum, and I tried it for the peperomias, and we found that it's a good media for propagation because it's very heavy. We also used sub-irrigation for propagating the leaf cuttings, since in the past we were doing it under mist conditions, and even though we're using perlite at that time, we still got a lot of rotting. But with that new system, we're doing it indoors and the water is underneath, and we have almost 100% success and no rotting at all under this system. So this is just an example of a setup just using food containers and turfes. So we just fill the food containers with turfes and then fill it up with water up to about halfway, and then we put the peperomia leaf or stem cuttings in there, and um, it's lighted under a grow light or under fluorescent lighting, and we get success with propagation. So in addition to that finding in terms of using the right media and then using the right environment, we also examined the use of rooting hormone or Hormex-3 um, in these cuttings, particularly Peperomia sandwichensis and Peperomia mawiensis. So we found that the rooting hormone actually increased the number of roots and also the root length of cuttings for both of these species. So rooting hormone was beneficial. We also tested single node cuttings with leaf. These particular cuttings are faster in terms of generating new shoots because you have a bud that's in there. And the application of the rooting hormone 3000 ppm IBA increased root numbers and root lengths as well. So in addition to macro propagation, we're also looking into tissue culture studies. And we did a bunch of, so earlier this year, we did some testing, just preliminary testing of explant sterilization using whole leaf, leaf section, single nodes, plus one leaf, and then single node without leaves for Peperomia sandwichensis and Peperomia mawiensis. And we tested different types of sterilization methods. We did, but the most effective propagation sterilization method was soaking it 1 hour in distilled water and then 5-hour sugar soak. Then after that, soaking it for 1 hour in 0.2% Physan plus Tween 20, and then 30 minutes under 5% Clorox and Tween 20. So this was very effective in office-grown plants. So we have plants that are grown indoors just to reduce the amount of contamination. And we got some success with that. So we have some cultures in, we have some leaf cuttings and then stem cuttings with shoot proliferation. Just last month, we tested a bunch of explants under greenhouse conditions, and I'll update on I'll do an update on that next year. Next would be the indoor trial update. So we're testing 2 different species. So we found Peperomia mawiensis and Peperomia sandwichensis as 2 species that have potential use as indoor plants because these 2 species have really nice coloration. They're compact and they're small enough for small spaces. So this is an ideal plant for something like an office type of house plant. So right now we're testing these 2 species under different light levels, so about 5,000, 750. So 750 is the office light, and then the 350 is about half as much of the office light. So these are pictures of the plants 5 months after installation. So this is Peperomia sandwichensis. You have the highlight treatment, the office light treatment, and then the low light treatment. Under the highlight treatments, the plants retained its red color. There's not much stretching going on, but in the office light and low light, there's some amount of stretching, but very slight, and red coloration slightly disappeared. So far, so survival has been very good, so we can say that this species can tolerate low light levels. The other species that we're looking at is Peperomia mawiensis. We installed them in May, and these are the pictures in highlight, office light, and low light conditions. So there's still good survival, although there's some death in the highlight treatment. Um, there's less red color again with lower light levels. And it seems that the Mauiensis stretched a little bit more than the Sanvichensis at lower light levels. Updates on the plant material distribution for testing. So we are propagating the Pawhiaka selection of Peperomia sanvichensis and Peperomia mauiensis, the EL3H selection. We'll be increasing that for the past year using stem and leaf cuttings. And earlier this year, we, in collaboration with Russell Galanti, we did a seminar at the Pearl City Urban Garden Center and gave away some plants to interested nurseries on Oahu. So with this seminar, we distributed 50 P. sanvichensis plants to Happy Koi Nursery, about 100 to Plant Hawaii Nursery. And then in February, we also distributed 6 Peperomia sanvichensis plants and 25 P. mauiensis plants to Maui Native Nursery, and 6 plants to 6 P. sanvichensis plants and P. mauiensis plants to Maui Nui Botanical Gardens. So some feedback with Peperomia sanvichensis. Some nurseries, so 2 nurseries have mentioned that it's a bit slow growing, but it's expected for a native. And 1 nursery actually started selling them at $25 each because they made it was on a custom made pot, so they increased. Originally, they were selling other plants for $20 in that custom made pot, so they just increased the price to $5 with this new peperomia. And they said that they sold out 1 dozen, and most of these are bought by native collectors. Later this, so the next month, on November 18th, we will be doing some market testing of Peperomia mauiensis in collaboration with Hannah Lutgen, who's the extension agent in Maui. So we propagated about 50 plants in 2-inch pots, and we will be giving it away for free with completion of a survey. So we developed a market survey to see how consumers will accept this species. So that's the update on Ala Vainui. So the next update is on Pawhiaka. So Pawhiaka is a endemic herb, viney herb that grows along the coast. When we did experiments in the past, so the last 2 years, we collected a bunch of accessions from the different islands like Oahu, Maui, and Hawaii. We saw some differences in terms of hairiness, leaf shape, and also branching habit. And these characteristics allow us to select plants or accessions that may be useful as ornamentals. So we've tested in past updates or reports. We did container plant evaluations of these species. These species is very easy to propagate from cuttings as well. And in past updates, we also gave you some results in terms of propagation. Based on research that was done 2 years ago, we finally selected 2 promising selections for release. So one would be Lion Arboretum, which is a white flower, and green leaves and purplish stems. So it doesn't have any pubescence, but it's a very compact growth form. And then the right picture here is Puhala Bay, which is a hairy leaf accession with which has a very unique wavy margin to its leaves. It also has pale blue flowers. So focusing on the Puhala Bay accession, so this is what Puhala Bay looks like in a landscape setting. It's a very dense growing Jack Montia and has nice blue flowers. And this is the same accession in a pot. So in terms of updates with Puhala Bay, we propagated 21 plants for testing in a residential landscaping situation. So Alberto Ricordi, which is who's a designer, a landscape designer, contacted me and was interested in testing this species or this selection. And so I propagated about 21 plants and I gave it to him. And he'll be giving me updates in terms of the performance of the plants under residential conditions. And probably next year, we'll be planning to release this accession jointly with the Maui Botanical Gardens. And the tentative name would be Puhala. So if you are interested in testing these selections, let me know. And I could send you some plants for testing. And then I'll ask you for feedback. Other exploratory projects that I'm doing right now is, so one would be the use of the flowers of Maoli Oli, which is a Shidia species, as a filler flower. So I've done preliminary base life testing on this species, and it looks like it can last for about 1 week. So I'm increasing these materials for now and trying to do further testing on base life. In addition to this, I'm also looking at propagation of variegated naupaka, seeing whether we can get many plant materials with shorter cuttings. So you'll see more updates on that on these projects next year. My talk is very short, but yeah, if you have questions, let me know. So I'd like to thank my student help, the different agencies that are collaborating with me, the extension agents, Russell Galante and Hannah Lutgen, and also my funding sources. And
You have a question?
if you have questions, let me know. We have lots of time for questions.
Thank you, Dr. Baldos. So, with your work that is going on in looking at these different native plants, it seems like there's a whole lot of material out there that perhaps remains to be discovered yet. Are there certain areas that you are targeting, such as maybe coastal or higher elevation plants that you're looking for?
So for the indoor aspect, so I'm looking specifically at peperomia species. So those are found from almost sea level to about, I don't know, like 4,000 feet above sea level. So we've been collecting peperomia species in that elevation range. And then for the landscaping portion, so things that are outdoor, we're focusing our collections in the coastal areas because that's where most of the houses are or developments are.
I see. And so you mentioned that there was one landscape designer who had requested plants from you to try out in his design. Does it seems like the landscaping industry, with the need, especially for state projects, to include native Hawaiian plants, would be, there would be a lot more interest from that industry to develop more landscaping material to add some variety to their designs. Is that something like the industry is reaching out to you about?
I've been with the Lich conference. I've been giving them updates on what I'm doing right now.
Mhm.
So yeah, they're in the loop in terms of what research I'm doing.
Okay. Oh, great. Well, thank you very much for all your work, and it looks like you've got a whole lot more to continue on, and I doubt you ever run out of material to look
Yeah,
into.
there's a lot. Yeah,
Thank you.
thank you.